We are now recording, and thank you very much, Alex, for agreeing to give a Build a Cell seminar, and welcome to the first seminar of 2023. And it's all yours.
Thank you so much. Can you actually see the slides?
Yes, we see the slide, but you're not in a screen sharing mode yet. Sorry, you're not in a full, now you're in a full screen.
Now
Perfect.
we are. Fine, super. Well, thank you so much for inviting me, and thanks to everyone wherever you are on the planet that you are zooming in. I do remember from teaching on Zoom the experience of talking to a computer, so I actually have another screen where all the Zoom people are, so if you, at least some of you could turn on your camera so I can talk to people, that would be highly appreciated. Okay, thanks a lot for inviting. As you instructed me, I should say a few words about who I am. I'm a professor in medicinal chemistry in Denmark, and the trajectory that led me here, it goes from MIT, Cornell, and then the University of Melbourne. And in those three universities, I've been doing drug delivery. I've been doing polymer chemistry, supramolecular chemistry, and yes, different facets of drug delivery. And in Denmark, I am teaching medicinal chemistry, and I suppose this will come to show when you see the presentation that the inspiration for me to build the artificial cells, to build a cell, it actually comes from nature, from just how awesome the natural cells are and multicellular organisms. This is what I learned at the synthetic cell, at the SynCell conference in 2022. We draw inspiration from very different places. Some of us, when we talk about build a cell, about synthetic cell, some of us try to make mimics of a structure of a synthetic cell. These are all the images I pulled shamelessly from internet. I hold no copyright for any of these. These are just some of the representative images I could find. So some of us build structures that mimic cells. Some of us pursue functions, such as, for example, encapsulated catalysis shown here. And indeed, some wonderful examples have been already published and more on the way. And again, as a chemist, as a medicinal chemist, my view on all of this, be it structure, be it function, is that at the end of the day, I see molecules. I see molecules that make all this up. And I am wild with how molecules work in nature. And I love to mimic this in synthetic cells. And specifically, what I'm mostly fascinated with is the notion of signaling in nature, because cellularity originated on this planet very early in the evolution, about 400 million years after the birth of the planet. And thereafter, the life develops as a cellular form, and in the multicellular organism, or even the single cell organisms between each other in the ensemble, they need to communicate to each other. They need to sense environmental cues. And what happens is that we have special molecules that typically sit in the membrane of a cell, and these molecules are called receptors. They perceive the signal that comes to the surface. Chemical messenger molecule does not enter the cell, it's the signal that enters the cell. And this notion of signal transduction, this sending of information through an otherwise sealed membrane, is what fascinates me so much. And then inside the cell, once you perceive the signal, there is all sorts of signaling cascades that actually end up with the response, be it cell division, be it non-division, be it moving, be it metabolism, etc., etc. And there is different types of receptors, and they're all absolutely fascinating to me. So when I talk about building a cell or a synthetic cell, what I would love to do is I would love to mimic nature. I would love to mimic life on the molecular level. I would love to mimic receptors and signal transduction on the molecular level. These are the things that I would love to do synthetically. And this is what I'll present to you today, our artificial receptors and our artificial signaling cascades. With regards to the word receptor, I really wish that the inventors of the English language came up with a few words that have different meanings. But unfortunately, the same one and the same word, a receptor, means many different things and refers to many different molecules. And the unifying common denominator is that all of these molecules are membrane bound, and they perform different functions such as recognition, signaling, internalization. So if I could, I'd name signaling receptors and internalizing receptors with two different words, because they're very different, but we're not at liberty to do that now. The first thing I'd love to present are artificial receptors in mammalian cells. I can tell you now that it turned out that making artificial receptors in live cells is much easier than making artificial cells in fully synthetic cells. So this is where we'll start. What we wanted to do is we wanted to make artificial internalizing receptors that would work in the most challenging of the cells we know, the primary T cells. Why do we need that? Again, I do not hold any copyrights for any of these images. I just typed receptor-mediated endocytosis, and you will see all the images that come up. This is because drug targeting is the new norm of medicine, right? Think about antibody-drug conjugates, think about all the antibody recognition events, cytokine recognition events. We need to talk to cells, we need to deliver drugs in a very specific manner. So if I were to make engineered cells with artificial receptors, this would be ultimate specificity of targeting. Because if I make an artificial chemical receptor using a ligand that is not found anywhere else in the body, I can make absolutely wonderful bioorthogonal receptors. And to give credit to all the people who worked in this area before us, we're not the first people to think about it. There's been synthetic receptors well before us. Typically, those were showing to work in cancerous cells, and we all know these cells are with very changed metabolism from normal cells. And the other thing is that these artificial receptors were typically micromolar by potency, which is not exactly on par with biological recognition events. So this is where we came into play, and this is what we designed. Again, give credit to the people who were before us. The actual design of the scaffold is not ours. What we did to it is innovative. This is the simplest of chemical artificial internalizing receptors we've designed. What you see is a cholesterol not drawn to scale. It's just like natural cholesterol, only it has a cholesterol amine. And this molecule has an interesting propensity to be internalized into cells and then cycled back to the cell surface. So if to this molecule you attach a ligand, this ligand would be shuttled into the cell and outside of the cell, and this can be used as an artificial mechanism for internalization. In our case, as the artificial ligand, we use fluorescein for several reasons. One, the simplest is that it's a fluorescent molecule. We can easily see it, locate it, analyze it. But the other is that it's a hapten, it's a xenomolecule, right? Xenobiotic. We don't have that in our body, but we can simply buy or raise antibodies to fluorescein. And this becomes a very a bioorthogonal cognate pair with high affinity between fluorescein and antifluorescein antibodies. So we make antibody drug conjugate that has this catepsin cleavable linker, and to it we attach monomethyl rostatin E, a very potent toxin. And you can see that this pair affords nanomolar potency of drug delivery. It's a bioorthogonal receptor with nanomolar potency of drug delivery. And not only does it work in cancer cells, it works fantastically well in primary T cells. These are primary T cells. You can see it still is nanomolar potency. And this is a fantastic and a very simple artificial receptor compared to other artificial receptors that are based on proteins where you need cell transfection, selection of cells that are positive for your gene, etc., etc. Here, it's very easy. It's a molecule that is worth 1,000 Dalton in mass. The installation protocol is just adding a solution of this receptor, and the cholesterol spontaneously inserts into the lipid bilayer, and it works in nanomolar potency in primary T cells. We were very excited about it. And where we took it forward from the paper that came out a couple of years back is that we, first of all, went away from MMAE to MMAF. The whole difference between these two toxins is that MMAE easily penetrates to the lipid bilayers and has a very high bystander effect, so it'll kill the neighbor cells as well, whereas its close analog MMAF is not membrane permeable, and this is much better for cell-specific effects. And indeed, here what we do is we raise the antibody to fluorescein, and then we conjugate MMAF. And you will see, what you can see here is the therapeutic window. So this is your drug, which is almost micromolar, and this is the receptor-mediated entry of the ADC, which is nanomolar. It's almost 100-fold therapeutic window, which is very beneficial. But while this has been advertised quite well, I think the other attribute of receptor-mediated cell killing is something we were surprised and fascinated with, is just how much faster it is. You can take MMAF, enough of it, and it would actually kill the cell quite well with nanomolar potency. Only for that, you need 3 days. And for real-life application in real-life biomedicine, this is just too long. Receptor-mediated process gives us nanomolar targeting and nanomolar potency within 2 hours of cell culture. So not only it is cell-specific, it is also very, very fast. So these were some new data which we haven't published, but this is work in progress and we'll be submitting soon. The second type of artificial receptor, again, still in live cells, in mammalian cells. Now we actually wanted to do signaling. It will still have a therapeutic outcome, but it will be a signaling. What we want to design is a molecule that would be sitting in a lipid bilayer, just like receptors would. It would be analogous to natural receptors in that we have exofacial part for ligand capture and interaction. It will have an in-membrane mechanism of signal transduction, and it will have a secondary messenger that would be released into the cell and have a therapeutic effect. And what we came up with is based on our prior work on prodrugs. Prodrugs are molecular entities that are devoid of therapeutic effect, but upon an enzymatic conversion, the drug is being released or unmasked, if you will, and then has a therapeutic effect. We worked with prodrugs based on glucuronic acid, and these do not enter the cell because the lipid bilayer is impermeable to glucuronic acid. And this is why these prodrugs are non-toxic. And you can see therapeutic window again is about 100-fold upon an enzymatic activity will reveal the toxin. What we realized is that if we take now this prodrug, which is non-toxic, and we purposefully associate it with the cell membrane, what we'll have is a receptor, dormant, which we can activate by adding an enzyme to release the toxin, which will be our secondary messenger. Again, we can make an artificial cell, mammalian engineered cell with an kill switch on it. This is what we designed. You will see this is our molecule. This is MMAE in red. In green, this is glucuronic acid as a trigger and masking group. Because it's a polar group, it will sit on the exofacial surface of the cell. And then we added a lipid anchor such that the entire construct sits within the lipid bilayer. It is a dormant toxin, but when we add the enzyme glucuronidase, we release the toxin, and it can have a therapeutic effect. This is what we did here. We engineered the cells. They are glucuronidase knockout cells. We installed the receptor, and the cells are very happy. They within the broad range of our receptor molecule, which remains dormant for many, many days. So at least the 5 days we've studied it here. But as soon as we added the enzyme, we release the toxin, and you see that stain, we kill the cancer cell spheroid. Not only we can do it by the added enzyme, but we can also do it through a co-culture of engineered cells with normal cells that have glucuronidase, and they secrete glucuronidase. So then our engineered cell becomes like a true Trojan horse, cellular Trojan horse that brings the toxin to the co-culture. So these were the two examples of live mammalian cells with artificial receptors. They are chemical receptors. There's no proteins. They are based on chemical molecules. Because of that, they're very bioorthogonal, and this is why it allows us to have very specific effects on mammalian cells. Now we can move on to the higher challenge, and that is achieving transmembrane signaling in synthetic cells. And why is that challenging? Is that if we talk about, just we did with MMA, MMAF, we have cellular machinery as a readout. In a synthetic cell, we actually have to engineer this readout from scratch. So not only we need to engineer the signaling itself, the receptor molecule, but also the readout, if we want something more exciting than just to change the forests. This is the design of our artificial receptor idea. It's not very dissimilar to what we just discussed. We will have an exofacial ligand capture ligand. It will be linked by this lipid tail into the lipid bilayer, and we will have a secondary messenger, which is dormant in the structure of the receptor, but upon enzymatic activation, the secondary messenger will be released and go into the cell. Therein, artificial cell, synthetic cell. Therein, the secondary messenger will activate an enzyme from its proactive form, the chemical zymogen, into an enzyme, and that would be giving us the response. I'll stop much more on the nature of the zymogens a few slides later, so let's first talk about the receptors themselves. This is the chemistry we came up with, not much dissimilar to what I showed with MMAE. You have an exofacial highly polar group. In this case, it's a phosphate. It's very polar, and therefore it sits on the exo surface. We have a lipid anchor to keep it in. And as a secondary messenger, we chose an amino acid cysteine. You'll see later why. We will be talking a lot about thiol disulfide exchange, and the secondary messenger with a thiol is exactly why we chose it this way. Now, this is the paper which we were discussing just before this lecture with Kate with the revision round. So this is very long in revision, but I'm hoping to have it published very soon. The scaffold for this receptor is modular and scalable. We synthesize it on a gram scale. I'll be honest with you, upon a request from the reviewer. From the scaffold, we can go any way we want in as regards to the activating group. So here's a phosphate or glucuronic acid or glucose. So we can make receptors based on different activating chemistry that would give us multiplicity of responses should we desire to have multiple responses. And in each case, if we add the cognate enzyme, so phosphatase or glucosidase or glucuronidase, we release the secondary messenger. Minor caveat that it was not with the glucose, and we figured out that this lipid tail actually inhibits glucosidase. Remains a mystery to us why, but we decided to move on to signaling. And this is signaling. This is to visualize the receptor localization in giant unilaminar vesicles, and signaling we do in liposomes. Receptor remains dormant in the structure of the lipid bilayer. There's no signaling. There's no enzyme activation with the receptor itself. But as soon as we add the cognate enzyme, so phosphatase for alkaline phosphatase for the phosphate protected receptor, or glucuronidase for the glucuronic acid protected receptor, we see statistically significant and well pronounced enzyme activation, which reports to us on the signaling. Phosphate was the best candidate. It was the most potent. What I mean by potent is how little do we need to take of the molecule to have reliable signaling. Glucuronidase specific receptor was equally good in signaling, but required more of the molecule. And glucosidase is, well, we see signaling, but not as pronounced. In this case, what we think happens is just glucose is not a charged sugar, so it must not come out of the lipid bilayer too far for enzymatic activity, so it's steric sugar. What we're so excited about is that we have a biological stimulus, or at least biologically relevant stimulus for activation of the receptor. We have a biologically relevant output, but the mechanism of signal transduction is pure chemistry. So it is a chemical receptor, it's a chemical signal transduction mechanism. What we wanted to prove next was that it is actually a signaling event and not just diffusion. There's a lot of efforts, highly successful, wonderful in their own right, efforts in signaling in synthetic cells based on diffusion, where two population of cells, or two cells, they exchange solutes, and through that they communicate. This is indeed very important to achieve, but this is not how it works in a cell. If you just do it by diffusion, you, by far and large, negate the whole point of cellularity, right? Cells are defined volumes, and they don't allow solutes by free diffusion for signaling. So this is what we wanted to see. Do we actually have signaling by diffusion or by receptor-mediated process? And for that, we compare cysteine as a secondary messenger or as just externally added cysteine. Now, if you look at this, this is enzyme activation with just soluble cysteine. And if you take a lot of it, millimolar concentration, obviously you push through, you create a very steep gradient, and you have signaling. But if you go to relevant concentrations, biologically relevant micromolar, there's no signaling. And this is what you see here, zoomed in, there is no signaling with say 2 micromolar cysteine. But if we take it as a receptor, because it's already in the cell membrane, upon activation of the receptor, we see strong activation and signaling. So indeed, it is not diffusion, it is a receptor-mediated process. And the second proof to this statement comes from our experiments where we add thiol traps. So we add molecules that would covalently react with cysteine or any other thiol for that matter in solution, but the thiol trap cannot penetrate into the lipid bilayer. In this case, what we see with cysteine is that this is non-inhibited signaling at a higher concentration that I showed on the previous slide. And with the presence of inhibitor, we inhibit signaling. That means that indeed, like I promised you, thiol trap would react covalently with cysteine. With the receptor, this is non-inhibited and the blue trace behind it is inhibited, but there's no inhibition of signaling. And that means that the thiol trap does not have access to the secondary messenger. The secondary messenger is in the lipid bilayer, again, to postulate that it's actually membrane bound and transmembrane signaling. This is what we've been very much excited about, that this is the first, to our knowledge, example of transmembrane signaling where we connect enzymatic activation to enzymatic output. And one of the interesting applications here is perhaps in sensing, because we have an encapsulated enzyme that is protected from the outside environment, and we can activate it through a receptor-mediated process, much like it happens in cells. Now, to artificial signaling cascades. Why is this so important? Is because, okay, we looked at these membrane-bound molecules at receptors, but a lot remains to be done here. It's the intracellular processes that, for example, this is adrenergic receptor, receptor that responds to adrenaline. For signaling, for signal transduction, what happens is then a cascade of events, and you can see there's phosphorylation, dephosphorylation in many different places, and many several enzymes connected in the pathway. Well, to realize all that in the synthetic cells would be a very high challenge. There's a lot of enzymes, and each of them needs to be controlled, and the activity needs to be controlled, etc. We looked at, but this is what we want to do. What we look, though, is not the native phosphorylation, dephosphorylation, or deacetylation. The reason we chose the artificial receptor with the secondary messenger being a thiol cysteine is that the signaling cascades I'll be presenting to you, they evolve around the cysteine, and that is to say the proteins that have cysteines. It turns out that it's a very rich platform, and the scope of it is very broad. The reason we can work with cysteine is that it's a fairly rare amino acid, but for example, in cysteine proteases, the cysteine is involved in catalysis. It must be there. So if we protect cysteine into a disulfide, we stop catalysis. But if we now take the disulfide to convert back into a thiol, we activate catalysis on demand. So this is just like phosphorylation, dephosphorylation works in the human cell. We can play with the cysteine and activate, deactivate an enzyme through that. An interesting add-on benefit actually is while the thiol is protected in the form of the disulfide, it's also protected from poisons such as covalent inhibitors of thiols, be it uracetamide, malamides, etc. But reactivation is on demand when you need it. Cysteine proteases are important because, for example, these are your caspases. This is apoptosis. But there are many more enzymes that have cysteines, and not always cysteine is involved in catalysis per se. If you look at the kinases, by some accounts, 40% of kinases have cysteine in the vicinity of the active site. And often, if you, as you will see in a second, if you modify the cysteine, you actually stop the kinase activity. And why kinases? Well, ATP is energy and everything for us. Polymerases, unexpectedly, also have cysteine in the vicinity of the native site, of the active site. So all of these enzymes are our tools to make artificial cells, to build a cell with artificial signaling cascades. Some of the data I'll be presenting just came out in our recent paper. Some remain unpublished, but hopefully we'll publish them soon. So we made three types of zymogens. The one we call zero length, that's the one where cysteine is protected in the form of a disulfide with the shortest, that's why we call it zero length, shortest methyl disulfide. For other cases, we make disulfides with polyethylene glycol polymer just to increase the steric bulk. In this case, the active site is protected by a fairly large polymer. And yet in other examples, we actually made a polymer which is a polydisulfide. So not only the disulfide linkage is with the active site, but also between each of the monomer units. The reason for it is that then you can activate the polymer and therefore the enzyme by a disulfide exchange at any connection between these red blobs and not just at the active site. And we apply this technology to diverse enzymes, and some data will be shown here. So here's the first example of a B-enzymatic cascade. We have a kinase, creatine kinase, which we can make into a zymogen form through a disulfide chemistry. The enzyme is not active at this point in time. We can activate it through the thiol disulfide exchange. It becomes active. It converts ADP into ATP, and ATP is the energy source. But in the simplest form, we can quantify it and visualize by luminescence because luciferase uses ATP as a cofactor for its catalysis. These are the three types of zymogens we used: the zero length, PEG-based zymogen, and polydisulfide. And you see there's no catalysis. But with a small molecule, with just DTT, we can reactivate and we have catalysis. So that's the first example, a very simple B-enzymatic cascade. Now we can make a cascade a bit longer. We connect the citrate synthase, an enzyme which is in the Krebs cycle, to light, to luciferase activity, because citrate synthase uses acetyl coenzyme A and releases coenzyme A without the acetyl group, that is to say with a valuable thiol. This thiol can activate the kinase takes ADP to ATP. ATP is our energy and for cofactor for luminase, for luciferase. Citric acid citrate synthase turned out to be an incredibly potent enzyme. If you look at it over 60 minutes, we can visualize and register luminescence at an incredibly low concentration. So this is also very good for, well, to make cascades, etc. Maybe light is not the most exciting readout, but here we connect kinase activity to transcription. How is that possible? Well, ATP is not only energy, but it's also a building block, right? And when polymerases make nucleic acids, they use nucleotides for this synthesis, and ATP is obviously the building block for A. And so we have a zymogen that does not have activity. We reactivate it through thiol disulfide exchange. It starts to produce ATP, and polymerase starts to use it for transcription. With a zymogen, there's no RNA synthesized. With a reactivated zymogen, there is. This is already a fairly complex cascade, very much relevant for the agenda of building a synthetic cell. Now, to explain why did we do the disulfide zymogen, the polydisulfide zymogen, reason for it is that both for the zero-length zymogen and for the PEG-based zymogen, we protect the active site, and only small molecules can activate the zymogens, as shown here. But to make reactivation possible through an interaction with other proteins, we designed this antenna, as we call it, such that it captures chemical information in solution. Then the polymer depolymerizes through self-formulative mechanism. In the interest of time, I don't stop actually on the mechanism, but you can read it in the paper. It's a fantastic mechanism, almost like dominoes decomposing. And in doing so, we overcome the steric shield. The interaction occurs in the solution. Enzyme activation occurs at the active site. And if we focus on the green and blue traces here, there's zero-length zymogen and PEG-based zymogen. There's no reactivation by proteins. But if we have the polydisulfide, it can interact with other proteins that have cysteines, and these will reactivate the zymogen. And very interesting, if we have two enzymes, both of which have cysteines, and for both cases we have cysteine that is important for catalysis, what we actually observe is a fantastic zymogen interconversion reaction, where the donor zymogen loses the protecting group, but it does so through a reaction with the other protein, which acts as an acceptor of the zymogen. And so the red protein here goes from a zymogen to an enzyme, whereas the green one goes the other way around, from an enzyme to a zymogen. To the best of my knowledge, it does not occur in nature, but it's a very peculiar mechanism which could be fun to use in the build a cell agenda. The presence of polydisulfide also opened up an opportunity for us to engineer specificity of reactivation. After all, disulfide chemistry is absolutely fantastic. It's bioorthogonal, it's biorelevant, but it's promiscuous with regards to the thiols that perform disulfide exchange. How can we make it specific? Well, we engineered a zymogen where this lipoic acid polymer has affinity to the activator. It's a thiolated beta-cyclodextrin. And in that case, only in the presence of this beta-cyclodextrin in a very low concentration do we actually reactivate the enzyme. But then furthermore, cyclodextrins can be inhibited by steroids. And this is what we show here, that this is the zymogen. We can activate it with the cyclodextrin and it works very well. But in the presence of chosen steroids, progesterone, but not cortisone, we inhibit the reactivation again. So in fact, what we make now is steroid control over enzyme activity. Just like the cell does it, we have steroids are amongst the most potent activators or deactivators of protein synthesis and activity. And we establish that in a synthetic setting, in a synthetic cell, or at least in this case, in a synthetic, with synthetic zymogens. So where we are today, and this is my one summary slide to the build a cell community, is what we've made in my lab is we've made artificial receptors. These are molecules that sit in a lipid bilayer that would separate the inner volume of an artificial cell from the external volume. And these molecules are needed to make our cells responsive. To make our cells responsive, that we perceive information on the outside, and then through the engineered signaling cascades, we can connect in environmental cues to be it light, be it proteolysis. I showed some examples with papa enzymogens to transcription, etc. So we've actually made all the individual steps and published individual steps. So what we're now trying to make is make it into a coherent, large picture where all of this is in the same artificial cell. And I'm hoping to have all this done by SynCell in Minneapolis. I'm much look forward to the conference. I already bought the tickets. Kate, thanks again for inviting. And thank you all for listening. This is my group, wonderful, talented people. And I would like to advertise Denmark as a place to do science because we have very good opportunities to get funding. Please consider if you're young students, I have vacancies for postdoc. Thank you for your attention.
Thank you so much. That was an amazing talk. There are questions in chat. Can you see chat? If you could read a question before answering it, that's going to go on the recording.
First, we have congratulations with Happy New Year. And I think we should actually, yes, Happy New Year to everyone. Kill switch is fantastic. Have you tested the ligand specificity? Specificity, well, we have two of them. Right, so one of them is here. So this is fluorescein to antifluorescein, and obviously specificity here is it comes from just the fact that it's an antibody raised to fluorescein. So this is the trace which is ADC in itself in a cell without any receptor, and there is no toxicity. So this is there's plenty of other receptors on the cell, but this ADC does not recognize them, so there's no toxicity. The second
example. Can I ask one more thing? So actually, my question is the ligand specificity. So have you kind of tested some similar ligand? You mentioned glucuronic acid, and then yes, glucuronic acid has multiple different compounds that structurally very similar, including glucaric acid or some other
You're absolutely correct. But to our eyes, these compounds are similar to a natural, to an enzyme. They're very different. So glucose does not respond to, or rather, glucuronidase does not recognize glucose, and glucosidase does not recognize glucuronic acid. So that's the sugar chemistry in the body. So we've done this on the receptors that phosphatase does not activate glucuronic receptors and vice versa. So no, this is orthogonal. This is beyond doubt.
So that there's no other compound that interfere signal.
I'm sure there will be other inhibitors, that's for sure. But this specificity is so high that there is even isoforms for, say, glucuronic acid, bacterial glucuronidase, mammalian glucuronidase. They might have difference in recognition and potency and affinity, but again, say two sugars are not cross-recognizing.
That's fantastic. Thank you.
But that's not my achievement. That's nature.
Yeah, of course. I mean, but you found that whatever nature, the features.
Was there another question in your note?
So the second question is, so I guess your circuit, the downstream, actuation is basically killing. But I'm a synthetic biologist, and I wanted to always kind of develop new circuit by just replacing the sensor. So you now have the one sensor that responds to, glucuronic acid, I believe. Mm-hmm. And then if you change that sensor to some other sensor that detecting some other, signal, you could make other, kill switch that respond to other molecule or other signal.
Absolutely. Do you still see my slides?
Yes.
Perfect. So I agree fully with you that cell killing is, as such, rather boring because that's the one we wrote, and it's much more exciting to make something work. And this is what we try to achieve here. So with this slide I was showing, we can have at least 3 different protective groups on the same scaffold, and only imagination is your limit. And then we actually activate enzymes, right? So it's the same enzyme. It's the same receptor as I was showing you, that later on would actually perform all of this activation. So what we're doing today is this is the same receptor that we can activate the kinase, or we can activate proteolysis, or we can activate the polymerases. So that's exactly what we're doing. So if you're interested in that, I'd love to collaborate, because quite honestly, I'm not a synthetic biologist. I'd love to pick up the brain of the likes of you. But that's exactly what we're trying to do. And we've actually accomplished all the steps. So we have the receptors, we have the cascades. So what my students are busy doing now, and I really hope to do that by the time we're in Minneapolis, is to actually merge these two things together, that we activate, that it will be, the title of the paper would be receptor activated transcription.
Sure. I'm happy to talk more offline, but my interest is more biocontainment and kill switch side. And then I'm not the mammalian, synthetic biology expert, but I'm fascinated by your talk, and then I thinking about multiple different, avenue we could do together.
I'd love to talk. Yeah.
Yeah. Thank you so much. That's fascinating.
The next question is, what is the limit of the artificial signaling cascade complexity? How many can you express in a cell-free system at once? That's a great talk. It's a great question. We have not expressed anything just yet, right? So all of these enzymes we take from nature. We then do chemical biology, we do chemistry on the proteins to modify them, but we have not actually expressed anything ourselves just yet. How many can be put in a synthetic vesicle? I believe I seen a paper, I believe it was Advanced Biology, Advanced Biosystems, where they put maybe 6 enzymes into interconnected cascades, but maybe not controlled by maybe not interconnected. So in our case, we did 3, right? So we have citric synthase to kinase to luciferase. Maybe linear is already not necessary. Maybe we need branching and feedback loops and things like this. And this is very hard, but I'm sure we will be trying to do, because this is fascinating. I hope I answered the question. Do those signaling cascades need a membrane? Do you do it in liposomes, or can it work in bulk solution? It obviously can work in bulk solution. So what I've shown here, this is in bulk solution. They does not need the membrane at all. Now, the reason we do it in the presence of the membrane is to build a cell. And in my personal opinion, subjective opinion, again, to capitalize on this cellularity, we need that impermeable membrane. It can be done, not just it can. These are the results in solution, but I think it's very important to achieve all that in an encapsulated volume when it's receptor mediated. This is my holy grail, at least for this year, I would say. Do the cascades need post-translational modifications? Chris Dannett, I'm sorry, can you please clarify what do you mean by post-translational modifications? We do, well, we make zymogens out of them. So we take a cysteine and we then control it through the disulfide chemistry. So yes, this is, I suppose, post-translational modification, because otherwise we don't have an on-demand activation, right? If I just mix these 3 enzymes in solution, they will be working all together without actually controlling or talking to each other. To have it on demand, I need this element of activation, deactivation. So this is what we're doing. Could you please comment on the possibility of using molecules that are entirely unnatural signaling molecules in the signaling cascades? Like building a cascade responsive to a non-natural metabolite. How hard would it be? There's a fantastic field of science for tools of chemical dimerization, induced chemical dimerization. It's not us 20 years old technology, fantastic. This is still biomolecules of proteins that respond to think protacs and all that. So you can definitely have chemistry that would be completely non-natural to talk to the enzymes. So if you can, or medicinal chemistry where we can have enzyme inhibitors based on fully synthetic molecules. So what we've been dreaming is, we just don't have resources to do everything, is if we were to, instead of MMAE here, it can be, well, MMA is a natural product, but you can envision putting a drug that is completely unnatural. And that, I suppose, would be what you're asking about. It would be controlled, the cascade would be controlled by a synthetic molecule. So I think that's a technical difficulty, not the scientific. What's the limit of the chemical properties of the artificial signal? Is there a limit to how polar can it be before it cannot enter a membrane? Yes, this is a great question. So this is MMAE as drawn here, and MMAF would be differed by, so this hydroxyl is a carboxylic acid. And when we tried to make the synthetic receptor with a carboxylic acid, that did not work. So MMAF didn't work. With regards to synthetic receptors, as shown here, maybe I'll look at the chemistry. We've made it with a cysteine, and quite honestly, every time I give this talk, people ask me, why did you even use cysteine? Shouldn't that carboxylic acid here prevent its membrane localization? Yes, it should, but somehow it worked in this case. What we're doing now is also changing this from cysteine to cysteamine or other thiol-containing molecules to facilitate membrane localization. Indeed, this is a great question. If the molecule is too polar, it will not sit in the membrane. That is indeed the limitation. That's a very insightful question. Yes. Or it can be stuck if it's too hydrophobic. You're absolutely correct. There's a continuation. Yeah. Kate, I reached the end of the chat.
Thank you so much. I had a couple questions, but actually I think one of the questions was already asked. I was going to ask about the expression too, and it sounds like you answered that already. So if there are no more questions, I just want to say thank you again to everyone.
It's a great pleasure.
Thanks. And yes, congratulations and have a great day, everyone. And see you next week. Bye, and bye, Alex.
See you. Thanks, Kate. Hi.